Yeah, so I'll tell you a little bit about the arc at Ginkgo. I've certainly seen every phase of the company's life cycle over the last 13 and a half years, and talk about where we're maturing the business and mixing sort of tech, hard tech, software, and tools and biology to offer different products to our partners. Uh, those product areas fall in a diversity of verticals. Over the years, we have worked in pretty much everything listed on the slide, and I won't have time today to talk about all of them. Happy to talk afterwards or offline if I don't touch on anything that you're interested in. But in general, we got our start in industrials as a very non-traditional company that I'll touch on a bit in a couple slides, but really got into pharma and into agriculture later in the company's life as we built our platform. And then started to branch out into other consumer technologies as well. Ginkgo was born at MIT, as many of you may know. Yep, thanks. It was founded by three PhD students in BE, one in CSAIL, and one faculty member, Tom Knight, who you see wearing his bell bottoms, working on his master's thesis, which at the time was making a mini computer, as they called it. And have taken learnings from Tom and the founding team to set the core mission of making biology easier to engineer. So that has never changed for us. We've always wanted to develop better tools to work in a variety of microbial and mammalian hosts to make useful products for our partners. Those products could be small molecules, they could be peptides or proteins, they could be the cell itself as a product. Particularly over the last 10 years, as we've been able to raise capital in the company and build out our platform, we've faced a number of hard challenges when you try to generalize cell engineering and applying tools in genetic engineering and high-end analytics to understand what modifications you're bringing to bear to a cell type of interest when you want to make a new product for a partner. And also thinking about what the importance of scale is and when it makes sense to automate or not. When it makes sense to build your own software tool or use something that's existing off of the shelf. And for us, 10 to 13 years ago, very little existed for tools for us. And so we did have to be pretty scrappy as engineers and build a lot of things with our own hands. Another interesting piece to our history, so being an early stage synthetic biology company in sort of 2009 to 2013 did not make it particularly easy to raise money. And so we bootstrapped the company, and for the first 7 years of our existence, did not raise any institutional capital from traditional investors, but rather built up the company to around 40 people wholly on grant funding. And so through a series of successful continuations on SBIRs, through the National Science Foundation, through DARPA, through a large award we had from ARPA-E, we were able to start to test our hypothesis of automating some of the core protocols and steps in engineering microbial cells primarily to be able to engineer pathways and make molecules of interest for our partners. And so before we came to have the ability to raise from venture capital, we already had around 10 or 12 commercial deals, and we're delivering on technical milestones, and agreed to enroll as the first biotech company in the Y Combinator program. So some of you may have heard of Y Combinator running in the Bay Area, a really fantastic program that helps very young startups get great mentorship to set a business model, to learn how to think about raising capital, to think about learning how to scale a business. We looked very different, but at the same time, Sam Altman, who some of you may know now from his OpenAI fame and other public exposure, was just taking over Y Combinator and reached out to Ginkgo because we looked like we had that sort of angle of tech where we made sense, and some of the things that we were trying to scale fit with his vision. And so even though we were already a somewhat mature startup, we were able to take part in the program, and it really opened up a lot of opportunities for the company to think about how we could build out a more mature version of our Foundry vision to automate DNA synthesis, assembly, strain engineering, and then all the ensuing analytics that come with that. So over the last year and a half or so, this is a sampling of partners that we've had and have now at Ginkgo. So we organize into sort of three primary verticals and work with companies large and small. As I said, had our beginnings in industrial biotech, where we weren't well capitalized in the beginning. We could not wait on FDA timelines, and rather found a lot of success looking at the ability to provide a biological solution to replace a product in the specialty chemicals market, in food and flavor and fragrance, where there was a lot of pressure and uncertainty over how a partner would source a high-value molecule of interest. We could take, for instance, the plant source of a flower or tree that would be used to extract said molecule of interest, sequence the genome, and then infer with bioinformatic tools what enzymes were being expressed to give you that biocatalytic function to make that product. And then needed to build out the tool set to sort of recode and refactor those genes in a microbial host that we knew we could scale to the appropriate scale, and merge with existing downstream infrastructure for purification. After some initial success in industrial biotech, we really expanded there, built out a portfolio of companies that were working in food, materials, and starting to get into pharma and natural products discovery. 6 to 7 years ago, we made a very concerted drive into pharma as well as into ag. First, through our founding of Join Bio, a startup that, sorry, my bad, go away, thank you. That was looking to partner with companies like Bayer to be able to solve really big moonshot problems in nitrogen fixation, in antifungals, anti-pest with biological solutions. That ultimately merged into an entire business unit at Ginkgo when Ginkgo acquired the Ag Bio business unit of Bayer. In the pharma space, we look to take our learnings in automating biological design and analytics to be able to provide solutions in improvement of viral gene therapy capsids, as well as circular RNA therapeutics and some CAR and TCR cell receptor cell therapy-based therapeutics. In addition to some microbiome-based plays that you see in the bottom in the startup space that has seen its recent challenges, but we hope will come around. So as we've seen the evolution of the kinds of programs and products that we offer to our partners, we've started to sort of balance the left side and the right side of the slide here. So originally, Ginkgo's business model had been to do very complex end-to-end R&D programs where we would typically work on multi-year timescales to design, discover the content that we would need to sort of synthesize and refactor and then engineer into the cell type of interest, characterize and optimize biosynthetic pathways or the expression of difficult to express proteins, and then deliver those where we would license the cells that we engineered to our partner to make their product of interest with some sort of back-end value share, be it a royalty that we would share on the revenue that our customer captured, or a part of the savings if we gave a cost savings to an existing bioprocess that existed in the industry. And so with that model, the hope was, that overall we could build a base where we would have a portfolio of potentially not blockbuster deals, but deals that would give you that back-end revenue as you continue to invest in the platform and have that R&D engine where we would stick to that goal of getting better and better at engineering biology, but not vertically integrating. And so through that, sort of getting that portfolio of sort of high-end small molecule and protein offerings, Ginkgo has to date manufactured at scale and continues on a year-end basis over 10 different products, small molecules and proteins, biocatalytic enzymes for different industries, where we're able to manufacture anywhere between like the 50,000 liter and 200,000 liter scale, either directly with our partners if they have that sophistication, or at a third party. And so that continues to be a part of our business, but a part of our business that today is frankly challenged when you look at the sort of markets and R&D budgets being rather challenged in different industries. We knew that we needed to find ways to work more efficiently with partners, as well as give offerings that could be more readily accessed, both faster and cheaper. And so on the right side of the slide, we've moved into a variety of offerings that we call tools. So they're, offering the ability to use the automation that we have in-house to generate compelling data sets that a partner can take and make use with as they see fit, if they're training models, if they're designing their next experiment and just need to increase capacity for their ability to generate certain kinds of data. We have an AI function and an automation function that work very tightly together to be able to look at offering both internal and external closed loop solutions for generating data with highly efficient integrated automation that I'll describe shortly. And so with these tools offerings now, we've sort of balanced from these 2 to 3 year highly complex R&D campaigns to something more that could take 2 months or 4 months to be able to quickly offer a solution that an R&D leader wants to access. Over the years, we've also built out quite a bit of what we call our code base. So interesting strain assets in the microbial world, libraries of regulatory elements, receptors that give us the opportunity to do some asset licensing too directly, but these obviously have to fill very specific niches. And so on this sort of complexity and reproducibility curve, we're looking to push more of our focus into the right side of the slide. For us, we always think about scale, not just scale up though. So I think in the synbio industry and in industrial biotech, early on, 10, 15 years ago, scale up on the right side of the slide saying, can you actually manufacture hitting your price points, hitting your product spec was a huge challenge. Looking at the diversity of infrastructure that existed inside and outside of the US, and knowing that the field was starting to be able to access the production of molecules at productivities and titers that actually could hit viable cost points for commercial partners, but then saying, like, I don't have any of the pots and pans to make this. I don't know how I'm actually going to scale up. That problem certainly hasn't been fully solved, but there has been retrofitting of existing factory infrastructure, as well as just more sophistication in how to do this more efficiently as the biology has gotten better. If you can make 10 to 20x more products, you don't have to think about building a continuous process or a new factory. You can actually fit into existing infrastructure better. At experimental scale, I'll talk about this a lot. So how do you automate basic and complex protocols in molecular biology, in analytical chemistry, in sort of systems biology-based complex measurement of cells? And then at organism scale, really what I intend to convey there is there is really no one size fits all in microbial engineering specifically, where using non-canonical hosts, building tools in different kinds of microbial systems really can bring value to accessing a broader diversity of the kinds of molecules you can make. And so building that out over the years, to date, Ginkgo's worked with over 80 unique microbial hosts, which has been a tall order. And, if you look back 13 to 15 years ago, we would have loved to have said, we're only going to work in E. coli. But, we quickly learned that hard lesson that is not possible. And, you really can learn a lot from working with weird bugs. Moving on to laboratory automation, describe a continuum here of going from the most basic forms of instrumentation that we're all familiar using in labs, a PCR machine, a walk-up plate reader, through to what we've really tried to focus on at Ginkgo and building sort of third and fourth generation integrated automation. Task-targeted automation, I won't hit on much, but some of you may be familiar with like a TCAN liquid handler, a robotic liquid handler that is able to reproducibly and robustly move liquids for you. Boring but important when you're operating, complex protocols, when you want to think about starting to open up biological design space that you can no longer really handle by hand. There are different ways to approach scale, of course, and we've invested in the balance of that. A robot will not always solve your problems, and in some case, it's far more efficient and elegant to think about designing a pooled screen where you can use a sequencing-based output to look at millions or even billions of unique um, molecular designs. But that said, we have seen that there is high value in being able to have targeted arrayed data generation where you can interrogate a single genotype per well of a multi-well plate, or your culture format of interest. And so I'll touch mostly on class 3 and 4 here and what we've really focused on investing in at Ginkgo. And so here are examples of the category 3 automation that we've built over the last 5 or 6 years. So we've actually retired a number of our work cells. They actually run in alphabetical order and have these fictional characters as their names. So you see that we've already eliminated A, B, and C. D and E are going off on their way pretty soon. But overall, building out integration where you center typically around a single robotic arm that is able to address different landing zones at commercially available laboratory hardware. So typically anywhere between like 15 to 30 pieces of hardware. You can think of liquid handlers, plate sealers, plate readers, in some cases even a flow cytometer can go onto a work cell. What you really want to be thinking about from a process optimization perspective is where do I find benefit in automating? I don't want to integrate for the sake of it because these are rather expensive. These systems can range anywhere from $600,000 to $3 million. They can typically take around 6 to 9 months from design spec to sort of full validation into production. And then if your demand signal changes, you got to go start over again. And so retrofitting these systems is quite difficult, and it's something that we want to do very carefully when we see where the sort of breakdown of program archetypes is falling year on year for us. All to say, these systems allow us to think differently about how we design an experiment and how we can address greater diversity when we look at a library of enzymes. If we are trying to solve a critical bottleneck in a pathway that we are engineering in a new host, we would love to be able to run a high throughput screen that lets us look at 50,000 unique enzyme sequences in some cases, or possibly 2 to 3,000 with unique experimental inputs and conditions. And so with these integrations over time, we've been able to write software at the very boring level that lets new drivers come about so that disparate third-party software and hardware can talk to each other. Very boring, but important. That you can upload data and process it and have good quality control metrics, because when you get to that scale of handling 10,000 samples in a batch, you need cameras, you need different logic for liquid level, you need different logic for transfer success validation, because you just can't get your head around that. And very importantly, you also need software that remembers what sample was in what well, what's the genotype of my strain in well A3, what reagent did I add to it at what time. And so we had to write all of that logic out that let these work cells typically replace the work of anywhere from 6 to 20 people in a given week. Now, we didn't fully replace those people. Those people became very good at working with software and automation, and it let them think differently again about how they would design an experiment. And so for us, oftentimes, we could then kind of push the envelope in how we would introduce diversity, be it combinatorial mutagenesis in a library of proteins, looking at combinatorial libraries of regulatory parts for driving gene expression, and running them on these systems at scale. Also, importantly, develop these both for microbial hosts and mammalian hosts, and added the needed containment to work with iPSC-derived cell types, primary patient samples, and doing different new workflows that we're offering to our partners in functional genomics and antibody developability in high-content imaging. And so overall, feel very comfortable when we're in that rational arrayed world of handling one genotype per well, of working with 10 to the 5th cell designs or experimental conditions. So then on to sort of generation 4 of integrated automation. We are developing internally a technology that we call racks, or reconfigurable automation carts. We believe that these systems get around those design headaches and stress that I touched on before, when I don't know if I'm designing the perfect work cell for 6 months from now, but it's going to take me 6 months to build it. And so here the idea is that we take laboratory hardware and enclose it in a reconfigurable automation cart. Uh, there are interlocks that allow this cart to merge up to a maglev rail. And so with that, what we can do is essentially have a Lego set for liquid handling robotics, for analytical systems. And so the idea is that we either have these on wheels where they come up to a rail, or in other cases, we have a sort of more streamlined version we call tiles, which can go into a tissue culture hood. We have a partnership with a national lab, for example, where we're putting these into custom anaerobic chambers, and we're able to run culturing and analytics with strict anaerobes. And so just thinking about like the sort of flexibility or the scale that you want to move to with these systems, we can offer that to our collaborators. And then in-house, we have a fairly large system that we continue to add to and refine, where we're able to run a large library of services for sequencing prep, for setting up digital PCR, RNA sequencing, cell cultures, where we're able to use our in-house software to interleave different protocols so that we're making the most efficient use out of this hardware as possible, in the footprint of our lab space, in truly a walkaway manner for many of these protocols. And so, racks collect into these rack systems. This is a rendering of another real system that we've built with a partner that lets us run large batches. And again, importantly, multiple different protocols, thinking about how to sort of be that train conductor and having everything run in the most optimal way so that you're making the best use of your hardware. We continue to do all of that boring but important work of bringing on new hardware so that we can have new capabilities and protocol steps that are supportable. And so you can go from a simple system to an entire room to be able to sort of run multiple different protocols. We've really found like a sweet spot thus far is in these sort of like 10 to 20 racks can serve, tens of related but diverse protocols for you in the lab. And then importantly, I touched on the software, but it's really important to make sure that critical steps in a protocol run at the right time. You're not seeing sample degradation. You're running everything together in the most efficient way possible. And so the logic here can simulate different scenarios of how you carry out a protocol, finds that optimal path, and then can recommend if you're running into a bottleneck, and we'll say, no, you cannot run this sixth protocol. These 5 have to finish first. Or maybe you need to consider solving that bottleneck by putting another rack unit on the rail. And so the nice thing there is that what we can do is essentially take off and put on different unit operations in the matter of a day. And then, a new unit op can be typically validated and vetted in 3 to 4 weeks. So this has really solved the problem that we've faced over the last 10 years when it comes to trying to make that bet on the big capital investment you're making and saying, am I making the right call on the science that I need to support? So what does that all boil down to? So, with the ability to still support these flexible R&D workflows, but offer them in a more lockdown automated service environment, we've greatly expanded the scale that our Foundry can offer. Our requesting program teams from our partners in our internal work. And so, wall of numbers, but in general, we tend to sequence everything. We tend to be able to prepare, very consistent inputs for metabolomics, for proteomics, for transcriptomics, to be able to answer questions on what a cell engineering plan has done to alter the metabolism of cell. the gene expression and sort of general overall state of said cell in the context of the process that we are going to either screen in or ultimately manufacture in. And so with that, that's let us build out a set of tools and approaches where we sort of have a fairly open playbook when it comes to solving the challenge of a complex metabolic engineering program. And so typically, we are living in a world where we're continually creating bottlenecks. Typically, you'll have your problem enzyme. It's one step of your pathway. Once you solve that, another one peaks out. You may see toxic intermediates. You may see, things sort of going in the wrong way from a metabolic flux perspective. You may see growth issues when you're trying to, run in a fed batch fermenter and produce at scale. And so, typically for us, and this is an example of one of those more traditional sort of long campaign R&D programs we're running for partners. We'll typically do rounds of different forms of metagenomic enzyme discovery and pathway design that will lead to a first demo strain. So we're getting that first confident evidence that we're making that first drop of the molecule of interest, but we know we're nowhere near to hitting our targets to be able to make things at scale economically. And so after that, then, we try to be good statisticians and design experiments that let us look at process parameters in context with what's happening with a given genotype of a cell design that we're working with. And so there, typically, we're looking at 2 to 4 month design build test cycles that will get you incremental improvements, as well as with the balance of sort of targeted arrayed experimental design and more unbiased ultra high throughput approaches. We have encapsulation and screening approaches, different sort of biosensor-based readouts to be able to look at a given genotype that we may not have inferred would perform better in this process. But again, typically you're looking at these sort of iterative cycles of getting to that point where you feel good about putting something in a big tank and spending a lot of money. I talked about the variety of microbial hosts that we work with. While I, quoted that number 80 hosts that we've worked with when it makes sense to work with a non-canonical host, that said, you can get a long way with a smaller subset of workhorses where, the name of the game is really to generate a broad and useful set of tools to be able to engineer those cells, understand how you're integrating into the genome, characterizing landing pads that give you stable expression, and do that again, all in the context of knowing how that cell is growing in a relevant fermentation process or bioprocess. Some examples here as we sort of prototyped parts and cell backgrounds. E. coli still is a workhorse for us. In general there, for making enzymes and screening enzymes, it's usually a very good first test bed. Unless we know something very specifically about like a complex class of enzyme or protein, or we'll immediately go to a new host. Pichia, also a favorite host of ours, and we did a lot of sort of pooled ultra high throughput work to evolve strains that were able to move away from traditional methanol induction. So Pichia is a very good protein producer, very friendly in different industrial contexts in general. But the canonical way to turn on expression of your gene or genes of interest was to spike in methanol. And you really don't want to have a 10,000 gallon tank of methanol sitting next to your factory. And so being able to move away from that, but in a way that the field had trouble with actually hitting that benchmark on being able to induce and express at the needed titers and productivities was difficult. But now we can offer our partners a set of production hosts that exceed the sort of that canonical baseline by about 20-fold. So being able to make industrially relevant proteins at 25 gram per liter is a big achievement and something that we've been able to offer a number of partners to be able to get proteins in their hands to work with. I'll leave it at that, but again, going into very complex fungi, sometimes that is the route to go. Sometimes looking at harder to engineer microbial species in the bacterial world, like Sautilus and others, can be the answer. And so, a couple of brief case studies here where we were able to sort of put this tool set to bear to good effect for partners fairly rapidly in many of these cases. So, early on in the pandemic, as vaccines were getting rolled out, there was a lot of concern over the availability of critical processing enzymes, and one of those being the vaccinia capping enzyme that was being used to finish off processing of the mRNA vaccines. We work together with a partner, Aldevron, that you may be familiar with, very good at manufacturing reagents, and quite worried that they weren't going to be able to supply all of their providers. And so in this case, in an E. coli process, by doing a systematic combinatorial layer screening of different promoters, RBSs, and sort of recoding of the subunits, we were able to, in very rapid order, get over a 10x improvement in titer, put that in their hands and get that scale to production that satisfied their demand. Also helped with Moderna and other partners to help in process improvements for making templates and other pieces in the vaccine manufacturing need and response. On the protein side of things, again, I already touched on that next gen expression system, but in general, what we've now found is that in these hosts, we can typically get to tens of gram liter titer for different classes of protein and enzyme at those very large scales, and use our small scale automated fermentation systems and even micro titer plates to predict which cell designs will give us those viable titers and profiles. And so here really starting to accelerate the ability to prototype a process and a cell design to get a specific protein of interest. Okay, so we can make a lot of protein, but is the protein going to do what you want and what you need? Typically biocatalysis can be a rate limiting aspect of industrial biotech process. Over the years, we've invested in a variety of computational modeling approaches, moving towards ML-based prediction of protein sequence that will give us expression, stability, activity, and continue to feed the data that we generate into those models to better understand how to improve different classes of these proteins. So we call this tool OWL. We use a mix of more traditional structure-based design tools that some of you are familiar with. We use the vast amount of data that we have put into our databases to do metagenomic sourcing, oftentimes finding an enzyme that may only be 30% sequence homologous to the known published enzyme, and gives us sort of a new starting point to protein engineer off of, with some chance of getting further improvements, since it's not the same protein that 30 very good labs have been looking at for a decade and trying to improve. And then thinking about how to feed that into different evolutionary scale models, both those existing out there and then feeding in our own data together with the assays that we run on our automation. Importantly, over the lifetime of Ginkgo, both through an in-house data generation and through acquisitions, so we're very fortunate to have taken on the assets and data of Warp Drive Bio, Loto Therapeutics, Radiant Genomics, Zymergen. We now possess a vast amount of data where we believe that if you compare it to the sort of scale of UniProt, we have around 2 billion unique open reading frames that have, in some cases, some characterized data where we have soil isolates that have metadata associated and have been sequenced. We can infer different pathway activities by looking at proximal location of different genes in relation to each other. And so we can feed that in and use that to drive better and better metagenomic prediction and find different starting points for our protein engineering workflows. And so to put that in perspective, as we run different rounds and generations of OWL, What we've actually found is that we can move away from scale as sort of a brute force tool to source a lot of things, but rather kind of work smarter and try to focus in libraries once we have a starting data set that we can work off of, assuming that we're within some viable range to the desired activity or other property of the enzyme of interest. And that actually fits really nicely with our rack automation because now we don't have these large monolithic automated systems that were optimized to run 10,000 samples, but rather we can run 10 batches of 100 samples on any given day, up to 1 or 2,000. And so sort of right sizing and not automating for the sake of it, but having that sort of flexibility to handle batches as you sort of respond to your data and have a faster turnaround on your analysis. And so some examples here that we've been able to demonstrate that this overall suite of computational tools allows us to rapidly find improved activities in some pretty hard to work with enzymes, P450s, phosphoketolases. You see the sort of fold improvement in activity in teal. Importantly, in gray, you see the library size. And so we find as we sort of focus and generate better data for the model to work on, again, we can sort of feed less design and give our partners a more cost-effective solution to a better engineered protein. I won't stare with the pathway logic for long, but just to say that again, with the scale that we're able to offer in sort of arrayed screening, we've been able to characterize a vast set of combinatorial regulatory logic that lets us have both inducible and non-inducible control over gene expression across orders of magnitude in a non-leaky manner. And so we can couple that when we have the challenge of pathway balancing or of looking at this, finding that sweet spot of expression for a protein of interest if any kind of downstream process challenges of concern or we have a toxic product that we're making. And so have a lot of control to fine tune once we've gotten to those sort of demo strains. Same goes for secretion control in various eukaryotes. So over the years have developed libraries of synthetic pre and pro signal peptides that help in the trafficking of proteins and ultimately their secretion to make sure that anytime you want that protein leaving the cell if the protein is the product because it's not having to do a cell lysis in your downstream process is fundamental to having an economically viable process that you can develop together with your partner. And so finally, like the strain background, so when it takes years at times to get to that final genotype where you've inserted various genes, you've deleted, you have added in chaperones, you've knocked out to have different oxytrophies. The series of getting to a healthy strain that can still be a viable production host is not trivial in many cases. And so now having this library, we're able to shave years off of projects at some point, and in some cases, basically drop in a new heterologous gene, optimize its expression, and go to scale. I'm going to skip over. Well, I mean, this was a good example for rapid work for a program, and this was translation from a mammalian system to a microbial system. So in this case, we used our picky expression system to engineer some known nanobodies for a partner who had basically cost concerns to be able to engineer at scale. And so with a fairly rapid design iteration, we're able to give gram per liter titers in a non-optimized fermentation process, switching from a mammalian system into Pichia. That got into our partners' hands and they ran with it. And then lastly, so making small molecules in microbial hosts can be quite challenging. Building up that code base and those strain assets does give you that good advantage to be able to engineer more quickly. We've, over the years, built out different pathway assets that allow us to make different classes of molecules such as terpenoids, phenylpropanoids, different phytochemicals relevant in ag. And so what we can do now is, essentially fine tune and tailor a given sesquiterpene project by mixing and matching different parts of that pathway and adding and subtracting those biocatalytic activities. Making sesquiterpenes at 30 to 40 gram per liter is very industrially relevant and something that we've been able to use to offer to a number of partners to be able to manufacture at scale, together with all the important chemical engineering that happens on the back end to extract these molecules that sometimes need solvent and distillation and the like. But good to see that some of these sort of canonical challenging pathway engineering problems in metabolic engineering have been addressed here. When we think about that, another good example, so typically you'll build up toxic intermediates, you'll have a mix of products when you have a 10 to 30 step pathway with different decorating enzymes. Um, Sort of built out the tool set where by using validated loci and landing pads and different knockout strategies, we're able to very rapidly, in this case, get rid of promiscuous glycosyl transferases, balance the pathways so that we can basically subtract all toxic intermediates and drive to a larger fraction of the product of interest to make your downstream more viable. And so, this was like a Herculean metabolic engineering problem that a lot of people have been thinking about, and happy to see that the combination of these tools can drive you to that successful product profile. And so, all to say, as we think about how we try to like methodically design these paths, what we tend to try to strategize on is optimizing on time when we are able to uh, properly size the way that we're spending on a program so that we can go on many parallel paths and then sort of combine the winning hits to get to a commercially viable strain and cell design much faster. And so, a program that maybe took us 6 to 9 years at the beginning of Ginkgo now is taking us 18 months in some cases because we can root through these and rapidly get to sort of a prototype starting point that can be optimized with the right tool. The last thing I'll talk about today, and then we can open it up for questions and discussions, is that idea of offering up different data packages directly to the partner. So in some cases, they don't necessarily need to have an engineered cell or protein in hand, but rather just want to see what comes out of a data set if they're doing different varieties of model training or they're designing their next experiment. And so Ginkgo has officially launched an antibody developability product offering, and where we've already started to partner with different biopharma partners, where we can essentially get a library of antibody sequences, rapidly synthesize the DNA, express, validate that the library of antibodies is folding and coming together. And then we run a set of sort of medium to high throughput analytical services to be able to look at just general QC of the production and titer, hydrophobicity, any aggregation or polyreactivity, various targeted functional assays. So we can, if there is a target that we can have addressable to look at recognition, in addition to general specificity, we can offer that data set to the partner, format it in a way that we can directly feed to them both raw data and curated data with metadata, and then have every step of what happened come from our LIMS into a translatable sort of pharma-friendly data package, if you will, for their own use. And so have nice validated sets and are expanding this now by an order of magnitude in additional to rolling out different offerings in functional genomics as well. And sort of seeing where the data as product is able to sort of empower partners to different ends. And so excited to have this rolled out now and see it already sort of picking up traction and growing. And so, the general idea is that, I touched on this concept of sort of closed loop data generation and experimental design. We're getting to the point where we have the infrastructure where, a request can be fed into our DNA synthesis and design pipelines. Those samples can be validated and then fed into our rack systems to be able to run a walk away, hands off set of protocols, not just one protocol. You can prep for many things, and then feed all of that data back into the appropriate data warehouses for processing and delivery, where we hope to be able to start to have that really start the next experiment as quickly as possible with the right validation and analyses. And so that's where we're structuring part of the business now as we sort of move through this challenging time in our industry, but proud of the technical teams at Ginkgo that have built this out and are keep offering that to our partners. So with that, I'll leave it to your questions, and thanks again for having me.